Good afternoon, guests, and welcome to our single-cell analysis webinar. My name is Declan, and on behalf of Oxford Global, I'd like to thank you all for joining us today. Today, Anjali, Seth, single-cell proteomics project leader at Cellenion, will be presenting on CellenONE, enabling proteomic analysis at the single-cell level. After Anjali's presentation, we will have a 10-minute Q&A session. Please submit your questions through the chat option on the bottom of your menu display. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the webinar. Without any more delays, I'd like to get started with Anjali's presentation. So, Anjali, over to you.
Hi, everyone. So thank you, Declan, for this introduction. So I'm Anjali Seth. I'm in charge of single-cell proteomics at Cellenion. And today I'm going to talk to you about our technology, the CellenONE, and how we can use it to enable proteomics analysis at the single-cell level. So first of all, a little bit about our company. So we were recently acquired by CellLink Life Science, who's the world leader in bioprinting. So their aim is to provide technologies, products, and services for biology. Our mother company is called Cellenion. It's the world leader in precision dispensing since 2001, and they are providing solutions for diagnostics companies. In 2016, they launched our spin-off, Cellenion, which is dedicated to single-cell research and development. Our mission today is to provide cutting-edge technology for precision single-cell dispensing. So we will act on different topics. So starting with single-cell omics. So we are trying to enable experiments like single-cell whole genome sequencing, single-cell RNA sequencing, nanoliter library preparation, also single-cell proteomics analysis using mass spectrometry. But we also have other activity that I will not go into deep detail today about cell line development for monoclonal antibody production, CRISPR-Cas9, stem cell development, and microbiology. What we want to achieve tomorrow is to provide ways to do single cell isolation as well as library preparation for all the omics purposes, and try to develop new pipeline for genomics, transcriptomics, epigenetics, and as well as proteomics. So our technology is based on pico to nanoliter dispensing. So how that works, so we have a glass capillary that is surrounded by a piezoelectric ceramic, and when we generate an electric current in the piezo, then there is an acoustic wave that is propagated through the liquid in the capillary, and that creates a drop. So basically the voltage controls the speed of the droplet that is ejected, and the duration of the pulse controls the volume of the drop. So what we can achieve is a volume from 50 to 800 picoliter per drop at a frequency of 500 Hertz. That means that we can generate up to 500 drops per second. We can work with quite viscous solution, and the precision of the volume is very high. So how we use that to do some single cell isolation? So basically our technology is image-based, which means that first when we load a cell suspension in the glass capillary, we have cells in our capillary, and then the software detects images of single cells. So what happens is that the software decides on an ejection zone, which means that that's the amount of liquid that will be ejected in the next generated droplet, and a sedimentation zone in pink, that is just the safety zone if we consider that the cell could possibly sediment. So what happens is if we see that there is no cells in the ejection zone, then the drop is dispensed in a recovery vial but is not isolated. If there is more than one cell in the ejection zone, same thing happens. And if there is a single cell in the ejection zone, but something present in the sedimentation zone, then the drop is as well discarded into the recovery vial. If we are in the configuration where there is one single cell in the ejection zone and no cell in the sedimentation zone, then the next droplet is going to be isolated. So if we look at real images of what we are able to see in our dispense capillary, you can see that exactly what I had said before. So something in the sedimentation zone then is not isolated. Multiple cells in the ejection zone, then it's not isolated either. If you have a single cell in the ejection zone, but more than one, or I mean, at least that the sedimentation zone is not empty, it's not isolated. But if you have a nice image of a single cell in the ejection zone and nothing in the sedimentation, then you isolate your cells. So just to let you know a bit more about that, I'll play a short animation of how the system works. So that's our machine, and you can see that if you go to have a look inside, you can work with up to 2 plates to undertake single cell isolation. It's compatible with well plates as well as design chips. You can work with 284 up to 1.536 well plates. This well plate can be controlled in temperature to improve cell viability. So first of all, it starts with the capillary that uptake the cells thanks to an acoustic technology. And then we can isolate based either on transmission images or fluorescent images, but just for transmission one. So you can see still the ejection zone that is in green and sedimentation here in red. So the safety zone. So what's happening if no cell is detected, then you generate the droplet in the recovery vial. And if one is detected only in the ejection zone, then you isolate. Then back to the camera. Again, a single cell is detected. So it's dispensed in the next well. Back to the camera. Then there is no cell detected, so the droplet is in the recovery vial. If you detect multiple cells, it goes as well in the recovery vial, which means that there is no sample wastage because you can then uptake again from this recovery vial. Single cell detected, nothing in the sedimentation zone. Then you have images of the cell where you can set up diameter, elongation, and circularity parameter. All images are recorded. Then if you get a doublet, it's not going to be isolated, as well as debris. And then we can isolate up to 40 single cells per minute. So this animation was obviously slow motion to allow me to explain a bit while he was playing, but if we see it live, it looks like this. So you can see that on the software, you have like a green button that shows you whenever a single cell is detected and isolated. So it goes quite fast. But if you have a doublet, then it's red, meaning that there will be no isolation. And we keep on isolating. So as I said before, the technology can be used either with bright field or with fluorescence imaging to choose on which parameter we can apply to isolate the cells. So we can work in bright field, but also with fluorescence and up to 4 different channels. So both the technology have their advantages. So obviously, if we work in transmission only, it will be very fast. We don't need to label the cells. The imaging is high resolution, and we can sort according to size and morphology of the cells. Because, as I told you before, we can set a parameter in the machine about diameter of the cells, elongation, and circularity to choose which cells are going to be isolated. If we base the isolation on fluorescence parameter, it will be a little bit slower, but then we can do many more things for the sorting. So for example, we can do live dead sorting to remove dead cells from subsequent library preparation. We can use specific marker subpopulation sorting, so isolate only single cell of interest if they have been labeled before. We can isolate rare cells. We can choose the best clones to achieve the clonal recovery and expressor levels, as well as isolating very small particles. So for example, bacteria that we cannot see in bright field, but we will be able to see in fluorescence. So now there is a short animation of how we can sort using fluorescence. So this one will show you how we can remove dead cell after a live dead staining. So for that, the cells that will fluoresce only in green will be live, but the cells that will fluoresce in green and orange channel are dead cells. So if we take up images, then we do the fluo, green, okay, orange, meaning that the cell is dead, so we discard. If it's green only, it's a live cell detected, so we then switch to transmission to record the parameters, and so on and so forth. So the video is as well slow motion now, so in reality it's 10 times faster than what you can see now. So our technology is highly versatile because we can isolate from 0.5 micrometer, so bacteria, for example, up to cells that are quite big. So for example, cardiomyocytes up to 80 micrometer. Our system is also very accurate. So the single cell isolation precision is very good. So if we see a batch of different cells isolated with the CellenONE, we can see that we achieve very high single cell dispensing. For different cell line, we have very few wells empty and even fewer wells with 2 or more cells. So just as an example, this is an image of cells isolated on a slide with a spacing of 500 micrometer, and you can see that when we can manage to achieve 100% of single cell isolation with 0% of the location empty and 0% of the location containing multiple cells. The viability of the cells are also very good when we use the CellenONE because the acoustic generation of the droplets is very gentle to cells, so the clonal recovery can be very good. So for example, if we isolate cells using the CellenONE for clonal recovery, we can achieve up to 95% compared to manual isolation. So now I will speak to you about how we can use the CellenONE to achieve nanoliter dispensing for library preparation. So before, as you know, library preparation are done using the FACS to isolate single cells and then well plates to do the library preparation. So this has advantages because it's very flexible, it can be automated, but it's also low throughput, has a lot of substrate limitation, and the reaction volume are quite high because they cannot be less than microliter. So every single preparation is quite costly because it's more than $100 per cell. If we use droplet microfluidics, then the volume can be drastically reduced, so nanoliter range. It's quite high throughput, but then one cannot exclude doublets completely, cannot do multiple intervention on cells, and you cannot link the cell morphology, so images, to the omics data that you will recover from the barcoded cells. If one uses the CellenONE to isolate cells in combination with custom-made consumables for very small volume, it means that there is no substrate limitation because in the CellenONE we can work with a lot of substrate and we can even design new one. We can work from picoliter to nanoliter reaction volume. There is multiple intervention possible because we can dispense cells as well as fluids. It's automation friendly and you can correlate the image that you get from the software to the sequencing data that you will get further the road. So it's good because then it means that you can know how a cell looked like when you get the transcriptomic results. Because the standard consumables don't allow us to work in small enough volumes, we have developed a chip that is called the Cellen chip, and we can work with 20 to 500 nanoliter in this chip. It's designed to have the size of a microscopic glass slide. And on this chip, there is 4 × 96 wells. So allow us to have 96 unique oligo-DT. And in one run, we can work up to 8 cell chip, which means that in one run with the CellenONE, we can have more than 3,000 single cells per experiment. So few examples of single cell omics application using our machine. So transcriptomics, genomics, and proteomics. So for the 3' single cell RNA sequencing, we have developed in-house a single cell 3' RNA sequencing workflow that is compatible with the CellenONE and our CellenChip consumable. So the barcoded oligo-DT are dispensed in the chip using the CellenONE. Then there is steps of lysis and RT buffer addition, and that helps us as well to rehydrate the oligo-DT in very small volumes, so 100 nanoliter. Then we can achieve a step of single cell isolation and cell lysis, always using the CellenONE. And then within the CellenChip, we can do the reverse transcription and template switching, and then pull the cDNA that has been generated by centrifugation. Then one can carry on with the amplification by PCR. So it means that if we use the CellenONE in combination with the CellenChip, it allows high performance with up to 7,000 genes detected per single cell. There is a low quantity of read needed. We can map up to 100,000 reads per single cell. It's suited for small samples, so from 100 to 1,000 cells. We have developed a simplified protocol that combines lysis and RT steps. It's minimally biased because we need a low number of PCR cycles. It's highly reproducible within biological replicates. The quality is high with minimal to null background. And it can be used with enzyme and reagents that are commercially available. Then, if you want to focus on rare cell transcriptomics, it's also nice to use the CellenONE because then you can work with samples that are very low volume and or very low concentration and recover more than 95% of your cells. So for example, an experiment that we did in collaboration with the University of California, San Francisco was to work on cerebrospinal fluid samples. And in those samples, there is very few cells, so 20 to 500 only in 20 milliliter. So if we use a centrifuge to concentrate a bit the sample to 20 to 500 cells in 5 microliter, we can then use the CellenONE to isolate these rare cells. And then once it's isolated, we can do the single experiment on those cells. If we want to focus on circulating tumor cells isolation and sequencing, it's also interesting because, as you might know, CTC are the seeds of metastasis and they indicate metastatic changes. So if we want to use liquid biopsy to investigate metastatic changes, it's minimally invasive, it's highly informative, and it's also dynamic because it's real-time monitoring tool. With the liquid biopsy, there is only 1 CTC per billion of cells present. And if we combine some CTC enrichment techniques, so antibody, magnetic size, or the electrophoresis sorting, to increase the number of CTC to 1 per 1,000 cells, then we can use the CellenONE to isolate the CTC and do some subsequent transcriptomic analyses. So now, the core of the webinar, which is single-cell proteomics. So this is a work that we did in collaboration with the Institute of Molecular Pathology in Vienna. So what we want to do is to provide a simple, easy, fast, and reproducible platform for sample preparation prior to liquid chromatography, tandem mass spectrometry, single-cell proteomic analysis. So what we want to achieve is to be able to use the CellenONE to isolate the cells, but also to achieve all the steps of sample preparation, as well as to miniaturize them in order to analyze the proteome. So what I'll present today is the experiment that we are doing now, and they are referring to the Scope2MS method. So it's a sample preparation method, and we want to achieve it using a single instrument, our CellenONE. So very briefly, because I'm not sure how much you are familiar with tandem mass spectrometry for proteins, a short recap of the technique. So tandem mass spectrometry is a technique that uses two mass analyzers that are coupled together to increase the ability to analyze chemical samples. So when it's used to analyze proteomic sample, it's a bottom-up technique. So first of all, the protein sample has to be digested into peptide fragments. Then these peptide fragments are going to be ionized, and the first spectrometer separates these ions by their mass to charge ratio. And then a particular ion from a selected mass to charge is selected and then split into smaller fragment ions. And these fragments are then introduced into a second mass spectrometer to be separated according to their mass ratio. And then they are detected. So the fact of using this second step of fragmentation allows to identify and separate ions with a very similar mass to charge ratio. Tandem mass tag are isobaric labels, so they have the same mass, and they are used in tandem mass spectrometry to enable protein quantification and identification. So tandem mass tag chemical structure is composed with an amino-reactive NHS ester group that will link to the amino group of the peptide. Then there is a spacer arm, and then there is a mass reporter. So when one uses tandem mass tag, it means that after the MS-MS fragmentation, each TNT label will generate a unique reporter mass in the low mass region of the MS-MS spectrum. So it's a little bit like using, it's totally like using a barcode. So basically you barcode peptide fragments with a TNT label. Then you do a first scan, you select the peak that you're interested. And then when you do the second step of isolation and fragmentation, we are able to detect which TMT label was on which fragment. And this is enabling us to quantify relatively proteins. So the Scope2 method works like that, with TNT labeling of single cell, and this allows to analyze peptides. So what's happening is every cell is labeled with a particular TNT label acting as a barcode. And so during the first scan, every peptide fragment that has the same mass is identified on the same peak of the mass spectrum, because at that point, all the TNT labels have the same mass. And then when the precursor ion is selected for further analysis, the second scan allows the accumulation and fragmentation, and then the fragments can be analyzed, and because all the TNT have a different mass reporter, it allows to say from which cells was coming which peptide fragment. So using TNT labeling allows isobaric labeling to multiplex relative quantification of proteins. So the Scope2MS workflow works like this. Now, as it is published, the single cell isolation is achieved with a fax, and then all the subsequent steps are done using a liquid handler, which means that you need to use two machines. So to do cell isolation, and then cell lysis, peptide digestion, and then barcode the sample before sample mixing. After sample mixing, the sample is injected into an LC MSMS for proteome analysis. So if we have a closer look at the sample preparation, it works like this. So cells are isolated with the FACS, then lysis is done, and then all the subsequent steps are achieved using a liquid handler, and volumes are in the range of few hundred nanoliters, which means that once you pull your samples from your 16 wells, because TMT labeling can be achieved with 16 cells, you will have more than 10 microliter per array. What we propose is to use the CellenONE to achieve all the steps of the sample preparation, so isolation of the single cell, as well as protein digestion, peptide fragment barcoding before the sample mixing. So if we have a closer look with the CellenONE, it should look like that. So a single cell can be isolated using our machine, as well as the steps of digestion, TMT labeling, quenching before sample pooling. So using the CellenONE, one shall be able to reduce the volumes from few hundred nanoliter to tens of nanoliter. So to reduce the volume for the sample collection to be at the end less than 1 microliter per array. So our first job was to be sure that this was going to be reproducible. So we repeated the exact same protocol as it was published in Scope 2 paper. And what we can see is that if we have a look at protein groups, peptide groups, as well as peptide spectrum matches numbers, the preparation that uses the CellenONE achieve the same capabilities as the one that was published in bioRxiv paper. So we are getting similar protein, peptide, and spectra identification number. We repeated that experiment 3 times. If we have a look at the single cell reporter ion intensity, we can also see that we are getting comparable sample losses throughout the sample preparation workflow. So the CellenONE achieve as nice result as the liquid handler when it comes to sample preparation. If we have now a look at the single cell channel reporter ion intensity correlation, we can see that it's highly reproducible. So every cell is detected nicely within 1 run. An additional experiment that we did was a cross-contamination experiment, because one could be afraid that using the same capillary to dispense different cells and then different labels could potentially have cross-contamination issues. So we did an experiment where we use dual proteome, so either yeast or human. We deposited with the single PDC. Then we did the workflow of sample preparation, TMT dispensing, etc. And what we can see from those results is that there is no cross-contamination within the samples. So we are safe if we use the same PDC throughout the sample preparation. So far, we managed to reproduce the Scope 2 experiment. So we validated that our machine can be used. So we can achieve the full workflow within 1 single instrument. And what we want to do next is to reduce the volume sample. So this should allow us to increase the enzyme to substrate ratio, increase isobaric label to peptide ratio, so reduce sample losses as well as reduce cost per cell. And soon as well, we'll launch a proteomic chip that will allow further volume reduction and that will prevent evaporation issues within the small volumes. And once all that is done, we'll launch a fully automated CellenONE workflow to work on the single cell proteomics preparation to have a really simple, easy, and fast platform. We are very happy to say that in the single-cell proteomics field, we are working with a strong key opinion leader in the field. So the IMP in Vienna, as well as Nikolai Slavov group in Boston. And we also have a CellenONE at PNNL to validate the fact that if they can use our machine with different substrate, the nanopods. The CellenONE can also be used for single-cell proteomics, but quite differently. So this is a work that has been done at the BAM Institute in Berlin. And in that case, the single cell was analyzed by laser ablation induced coupled plasma time-of-flight mass spectrometry. And the cells were arrayed using the CellenONE. And then the LA-ICP TOF-MS was used to look at isotopic fingerprinting of single cell. So what they did in this experiment is that they used two metal-based probes to identify either protein or DNA content in cells, but as well they were able to measure endogenous metal at the single cell level. So using the CellenONE to isolate and array single cells in combination with laser ablation ICP TOF-MS allows quantitative determination of analytes down to 100 attogram. So we are able to measure several endogenous metals at the single cell level, but also to use any metal conjugate antibody to multiplex protein quantification. So to have ionic and metallomics multiple parameter study at the single cell level. So to summarize, CellenONE is a unique technology for omics field. So we do achieve high single cell accuracy and certainty without having any doublets or any debris. We can work with any cells or nuclei up to 80 micrometers in diameter. We can use the CellenONE to do nanoliter dispensing for library or sample preparation because our system works with aqueous solution as well as organic-based solvent. We can work with rare cells because we need only few microliter of cell suspension to isolate, and we have very high recovery rate. We are able to visualize sample and record images of all the isolated cells. So this is as well really nice because we can correlate all the results that we get from the omics experiment to the images of the cells. So it's an open platform for new protocols. We are delighted to say that we have a lot of reference customers and partners. So a lot of prestigious cancer research centers, as well as people working in single-cell mass spectrometry proteomics, sequencing platform, people working on rare cells, as well as environmental samples, and a lot of partners for the region. I just want to let you know that in a few days, my scientific and managing director, Guillaume Tournière, will give a full presentation during the conference on the CellenONE. He will go a bit deeper into the results. And if you need any further information, please feel free to contact us. Thank you very much for listening.
Thank you very much to Anjali for this very informative presentation. And so now we move on to the Q&A section of the webinar. So as a reminder, please submit your questions through the chat option on the bottom of your menu display, and we'll work through them. Yeah, so we have one already currently. The question is, does the software differentiate different cell types?
So the way that it's used to sort cells, it relies on cell diameter, cell elongation, and circularity. So it's up to the user to set up the parameters to choose the cells they want to be isolated. Okay,
well, yeah, brilliant. Thank you for that. That answers people's questions. The next one is, what type of solutions can you dispense when using the CellenONE?
So that's the nice thing about the machine, is that you can dispense either aqueous solution as well as organic based. So as long as they are not too viscous, you can, in theory, dispense anything.
Okay, thank you for that. We have another one here. Could we have an idea of the price of the system?
For that, I would let you contact write an email to the contact, because it's the salespeople who are taking charge of that, and I'm not aware.
Okay,
I'm aware, kind of, but there is a lot of different options that you can get. So for the system working only in bright field, or the system having fluorescence and either one or several fluorescence channels, the price is going to be quite different. So according to what can be of use, I think it's better to contact them directly.
Yep, that's no problem at all. Brilliant, thank you for asking that. So a question here. The movement of cells towards the tip depends on gravity, so the sedimentation. What is the rate of cell dispensing? Was it 40 per minute? Question mark.
So yeah, actually the 40 per minute comes from the fact that we do have to dispense some drops in the recovery vials if they are not containing only one single cell in the ejection zone, and some or no in the sedimentation zone. So 40 per minute is the thing that we can achieve if all the parameters are set correctly. But I mean, it's not the frequency we operate. We operate at much faster frequency, but 40 per minute is the rate that we manage to achieve for isolating cells.
So we have our next question here. When we are talking about single cell proteomics, do you have a big issue of masking effects because of the abundant proteins?
Mm-hmm. So actually, that's why people are trying to move to single cell proteomics, because already with the TMT labeling, and it allows you to discriminate better between all the proteins that are going to be present in your cells, and even more at the single cell level. So this is not overcoming all the issues, but it's already improving the results a lot.
Okay, brilliant. So next question here. How do you perform the lysis for SCMS in scope 2? You need frozen/thawing cycles. How do you manage that with the CellenONE? For example, do you take the sample out of the machine?
Sure. So far what we did, I mean, what was presented today was just the repeat of the scope 2. So yes, in fact, for that experiment, we did take out the plate of the CellenONE, did the freeze-thaw cycle, and then put it back on. But what we are currently working on is to find the best lysis technique that will allow us to identify the most protein, and also to be lysis that is based on reagent dispensing. Like this, the workflow could be done completely inside the machine.
Okay, brilliant. Thank you for answering that question. So the next one is, what is the size, so inner diameter, of the capillary tip?
This depends. We do have several sizes. So according to, if you want to have very small drops to bigger drops, we can tailor that.
Okay, brilliant. Next question. Do you have a way to avoid sedimentation?
So we don't have a way to avoid sedimentation, but fact is, because the cells are in suspension, they are not sedimenting that fast. Actually, it's never been an issue. So because we are loading the cells at they are not so concentrated when we are loading the capillary. So I mean, the sedimentation is happening a bit, but compared to the rapidity of the dispense, it's not an issue usually.
Okay, brilliant. Thank you for that. Does it involve any cartridge with limitation on size of cells?
So we can dispense cells that are as big as 80 micrometer in diameter so far. We are working on ways to improve that, but no, we don't use cartridge. It's a glass capillary that can be loaded. So no, it's a capillary that can be reused.
Okay, brilliant. Thank you for that. Next question. Is the amount of proteins from a single cell enough for a single MS run, or does it use any spike mixture?
No, it's enough because actually the scope 2 is relying on the fact that we have to use a carrier channel. So one of the channel is actually composed of 1,000 cells approximately. So there is one channel that has 1,000 cells and the 15 other ones that has one.
Okay.
So that's the way they are doing it. So we just repeat the workflow in this experiment.
Okay, brilliant. Thank you for that. Next question. Can this dispense floating cells like adipocytes?
I think, I mean, I'm not the best person to answer that because I haven't worked with all the types of cells, but I think we do. I'm pretty sure we already did, even.
Okay, no problem at all. Thank you for that. So we have our next question here. What is the optimal cell concentration for dispensing cells at your target speed?
So I think on top of my head, it's, wait, I'm not exactly sure. I think it's 200 to 1,000 cells per microliter.
Okay, yeah, brilliant. Thank you for that.
But I would like to say that for all these technical questions, if people just want to write an email to contact, they will have a very precise answer.
Yeah, of course, brilliant. So have you tried to analyze smaller molecules with the MS?
You mean something else than peptides?
I think so. They've just put smaller molecules.
So I haven't personally. So my answer would be no. But then for the people who are using the technique that I presented for the work that has been done in collaboration with the BAM Institute, the fact of using the laser ICP TOF MS, yes, then they can analyze metabolites, I think, as well. Because they do analyze metals, so it's smaller.
Yeah, okay, brilliant, thank you for that. So can I have one more question? Does this involve any fractionation, and what kind of MS and MSMS techniques were used?
So we use the orbit trap, so the regular tandem mass spectrometry workflow. So I'm not quite sure. I mean, it's just exactly the same instrument and all that we used from the paper that was published on Scope 2. So we don't do anything more than they do, except changing the instrument that we use to prepare the workflow so far.
Yeah, okay. Brilliant for that. I have one here. What is the advantage of using CellenONE to isolate cells compared to FACs?
So compared to a fax, I'd say that first of all, because we rely on acoustic wave for dispensing, the system is gentler to cells. We can achieve higher recovery. And more importantly, even we can work with small samples. So for people who are starting with very few cells, fax is not really relevant, whereas they can achieve very high cell recovery from small sample working with CellenONE.
Okay, brilliant, thank you for that. Another one here. So what is the advantage of using CellenONE to achieve the sample preparation for the Scope 2 method?
So for now, as I said, like the best thing is that you can have one instrument only. So instead of having to use a fax and then a liquid handler, you can do in theory everything within the same machine. Obviously now, because of the lysis, you still have to take your sample out to do the freeze-thaw cycles, but we are working on that. So soon we'll be able to come up with a lysis technique that can be done inside the machine. So first of all, one instrument only, and then also the fact that the CellenONE can be used for other stuff than just dispensing reagents. So you can rely on the fluorescence and everything to sort out your cells before starting the preparation.
Okay, brilliant, thank you for that. So I think this is our last question, unless anybody else types any out. So how low could you miniaturize volumes when using the CellenONE for this sample preparation?
So now we did repeat the Scope 2, so same volumes. We did half of the volumes, so achieving approximately 1 microliter at the end, but in theory, because we can go very low with the drops that we dispense, even for reagent, we can go to the 10th of nanoliter only for the full prep.
Okay, well, thank you for that. So we don't seem to have any more questions, so that brings the webinar to a close. So as a reminder for everyone, Cellenion Scientific and Managing Director Guillaume Tournier will be speaking at our upcoming virtual Next Genomics series, taking place next month between the 4th and 6th of November. If you would like further information on the Congress or like a copy of the conference brochure, then please contact our marketing department or visit the conference website. On behalf of Oxford Global, I'd like to thank everyone for joining us today. I'd like to say a big thank you again to Anjali, who's put a lot of time and effort into making this webinar as interesting and valuable as possible. So I hope you all have a great day, and I look forward to welcoming you at one of our future events.